here, and we deleted the specific part of 50 basis region around this specific SNP. And we got that was in colon cancer cells, and we got the phenotype. Low cells, they were not proliferating, and we had a different type of, we entered here also the RNA, the transcriptomics, RNA sequencing, and we had about more than 600 genes that we were modified with this specific deletion of this part of the gene. So what we understand is that we open a new door using this multi-omics approach. We open a new door. We have a specific phenotype. It could be therapeutic, a target for new therapeutics. Understanding that a very small part, an intron, is not within the gene. It is an intron. And it is correlated to, it can modify the expression of more than 600 genes. So this is a very interesting approach. One of the most interesting examples that I know in the literature for this multi-omics approach is 10 years ago. There was a scientist from Scripps. You can see him here. This is the publication in Cell. You see personal omics profiling. He was able at the time to have access to the different technologies, so he followed himself. He first analyzed his genome, and then he followed himself for about 15 months. During that follow-up, he was able to make billions of measurements. You can see here 3.2 billion for the nucleotides of DNA, but also 3 billion of fluctuations of different molecules, proteins, metabolites, microRNAs, etc., etc. During that period, he had a virus infection, and he understood that there was a shift of his metabolism to diabetes. It was the first time that virus infection was correlated to diabetes. Diabetes is a lifestyle disease, and with specific diet and training, he was able to stop it and shift back to the normal. So the comment in science was that this will be the tour de force number one of therapeutics in the future. Talking about the future, you are familiar with this type of gadgets like smart watches or oximeters, etc. Utilizing different instruments, but think about micro-mass spectrometer. This spectrometer, this MS miniature is 15 centimeter long. And think about HPLC on a chip or GC on a chip. And think about that this is incorporated to a smartwatch. You can see that there is already different companies that they sell them. And you can see that you can have this kind of biosensory resonance-based biosensors that are integrated to this type of gadgets like a smartwatch, and can follow specific fluctuation of different metabolites on your blood. So this is how it works. This will be integrated with your smartphone, and you can follow your health every day. There are still a lot to be done, and mainly the most important to be done is this integrated, standardized data that we can apply from the different methods that we use. But it is for sure that the next decade we will have all this data integrated in therapeutics. And I'm not sure if it will work. I hope that it will work. The main idea is the holistic view. This is more than doesn't work. This is very interesting. The holistic view. Omics can give this holistic view, and this is the take-home message from this. And with that, I'd like to thank you.
Thank you, Professor Elliger, for your insightful presentation. Before we move on, I would like to inform you that after a short break, we'll have a Q&A session, so you will be able to ask questions as well. And now I would like to give the floor to Dr. Leo Rodriguez, an Omics project manager for Pharmaceutic.
Do you hear me well? So good morning everyone. It's my pleasure today to talk with you about Omics technologies as a new tool for integrating the microbiome in human healthcare. So I'm Julie Rodriguez. I'm Omics project manager at Biotech Research Institute. So my role is to study the microbiome analytical method and to try to standardize these new emergent technologies. And I'm also supporting the microbiome-based medicinal product developers in Europe. So Biotech Research Institute is a non-profit organization founded in 2010, and our mission is to identify and confirm the regulatory and scientific requirements for the development of microbiome-based medicinal products for the European market. And we count about 100 industry and academic members in this organization. So the microbiome's definition comprises all the microorganisms that live in an ecosystem associated with their theatre of activity, including the structural elements, but also the microbial metabolite that they are able to produce. The human microbiome is composed by trillions of microorganisms living in and on the human body. And human microbiome represents more genes than our own genome. The human microbiome is also able to produce hundreds of metabolites, and most of them with unknown effect at this time. So omics has emerged as a new technology that allows the exploration of this unknown world, this unknown part of this human microbiome, since to date there are many microorganisms that are unidentified because they are inaccessible by cultivation-based approaches. So it is important to note that the human microbiome is permanently interacting with its host, but also with the environment. It means that the microbiome from human interacts with the other microbiomes found in plants, in animals, in oceans, and so on. So there is a real interconnection between all these ecosystems, leading to the concept of the One Health concept. For instance, in the human body, we can observe that the microbiome composition is really site-specific because we observe a real different microbiome composition in the skin microbiome compared to in the gut microbiome, for instance. This is due to the different interaction that the microbiome have with their environment. And these differences are already present at the phylum level, because in the taxonomic classification we have different levels, and we already observe such differences at the phylum level in human body for the different microbiomes. Excuse me. So, for instance, regarding the gut microbiome, several studies have highlighted that several factors are able to impact both composition and function of the gut microbiome. These factors include essentially the life habits of the host, such as the diet, the physical activity, the disease, or the drug medication. And the different factors can alter and change the microbiome composition, leading to a manipulation in the production of the microbiome metabolites that can have an impact on the host physiological function. So, just to illustrate my talk, we observe a very large diversity of microbiome metabolites that can be produced in the gut microbiome, and these different metabolites can target different receptors, not only in the gut in order to improve the gut barrier and the immunity, for example, but also on the peripheral organs, because some of these metabolites are able to enter into the systemic circulation and to reach peripheral organs in order to regulate some key functions such as glucose homeostasis, fat accumulation, and so on. So how is it possible to study all these very complex interaction between the microbiome and the host? For this, we use obviously the omics technologies. So among omics, we have several available technologies. The first one is metagenomics that allow to explore the microbiome genome in the community. Then we can also use metatranscriptomics to evaluate the gene expression in the community. We can also use metaproteomics to evaluate the protein expression. And finally, we have access to metagenomics studies in order to identify and to detect the microbial metabolites in our community. So for the first one, I will start with the metagenomic approach. There are two different kinds of approach. The targeted approach, in which we focus in only one gene. So for example, in bacteria, we study the 16S rRNA gene sequencing. That is the gene coding for ribosomal RNA that is present in all the bacteria, but the genes contain 9 hypervariable regions that allow us to discriminate and to identify the different bacteria. So for this, we isolated the DNA from fecal samples in order to study the composition of the gut microbiome, and we amplify one of the 9 hypervariable regions. The most used is the portion between B3 and B4 region. For this, we amplify this region by PCR, and then we sequence this portion of the genes. And we obtain several sequence reads after the sequencing, and we process these sequence reads by using bioinformatic tools. So just to understand, there are two main methodologies to do it. The first one is to cluster together all the sequence reads that seem similar in order to create the operational taxonomic unit OTU, or we can just clean all the sequence reads obtained by the sequencer by removing the PCR errors or abnormal sequences. And then, once we have amplicon sequence variant or OTU, we compare this sequence with reference database in order to assign the taxonomic identity and to identify the bacteria. The other metagenomic approach is the untargeted approach called the whole genome shotgun sequencing. This time we are not limited to the bacteria, but we studied all the microorganisms present in the community. And this technology offers a higher resolution since we are now able to identify the bacteria at the species level, so the most detailed level. For this, we extracted the DNA from the fecal samples. And now we perform the DNA fragmentation and we sequence all the DNA fragments. And now, after the sequencing, we obtain a sequence read that we process differently by using also bioinformatic tools. So the first methodology is to align this sequence read with the known reference database in order to identify the microorganism present in the community. And the second strategy is to assemble the small sequence read into a larger sequences in order to reconstruct the whole genome of the microorganism. And with this technology, we have access not only to the taxonomy identity, but we can also predict the functional profiling, so the function in our community. But it is just a prediction at this level. So what can we do once we have assigned the taxonomy and we know the bacteria or the microorganism present in the samples? We can just compare the relative abundance of each bacteria between different samples using bar plot visualization. We can also evaluate by measuring some indexes, the bacterial richness and hiddenness in our samples. It means that we can evaluate the number of genus or species present in the samples with this omics approach, but we can also know the distribution of the different bacteria present in the samples in order to evaluate if the bacteria are equally present in the community or if some bacteria predominate in the samples. So in the examples, we can see the results for one cohort of obese individual, and you can see with the measurement of one bacterial richness in this, that the orange sample is more diverse than the red, the green, and finally the blue. And the last analysis that we can perform with this data is that we can compare different communities in order to see similarities and dissimilarities between the gut microbiome composition. So in this example, I transfer the gut microbiome from obese individuals into recipient mice. And you can see, for instance, in the blue group, that the mice into the four groups that receive the same gut microbiome are clustered together, so have a real similar microbiome composition. And these mice are similar in terms of microbiome composition with their respective donors. So this approach were for the composition of the community. Now for the function, for evaluating the function, we can use metabolic approaches. So for this, we can detect microbiome metabolites that are present in different specimens because we can identify microbiome metabolites, but we can detect them in blood, urine, feces, or even race. We use some analytical methods such as chromatography, spectrometry or nuclear magnetic resonance. And we can use here again two different kinds of approach. The untargeted approach for just comparing the metabolome profiling between different samples, but with this approach we have also access to unidentified metabolites. Or we can use the targeted approach for which we use a reference database with known metabolites in order to detect the metabolites of interest. So, omics enables the integration of microbiome in the drug development process because it allows to develop three kinds of application. The first application is that omics allows the identification of bacteria, consortium of bacteria, bacterial components, or bacterial metabolites that can be interesting for the human health and that can be proposed as new drug candidates. The second application is that omics allows to evaluate the impact of the microbiome on the drug candidate's metabolism. And the third application is that omics can also allow to evaluate the impact of one drug candidate on the microbiome composition and function. For example, for the first application, a lot of novel medicinal products have emerged on the market thanks to the omics technologies, and these products have the intention to treat, prevent, or cure in animals and animals through the modulation of the gut microbiome or of the microbiome. Among the different products, we have the whole ecosystem-derived medicinal products that are derived from complex microbial ecology. This is the case for fecal material transplantation used in the context of Clostridium difficile infection. We also have the rationally designed ecosystem-based medicinal products for which the active substance comprises simplified and well-defined microbial ecosystem. So this is the case when we do a mixture of different non-bacteria, for instance. We also have the live biotherapeutic products in which the active substance is a living microorganism, such as a bacterium, or a non-living biotherapeutic product. In this case, this is also a bacterium, but this bacterium has been intentionally and previously killed or inactivated during the process. And finally, we have the biotechnological product in which the active substance is a product of fermentation or a microbial metabolite. For the second application, several studies highlighted that the gut microbiome can really impact the response of certain drugs. This is the case, for instance, for the cancer immunotherapy, in which we observe that some individuals respond very well to the immunotherapy, whereas some other individuals did not respond. And one explanation remained in their microbiome characteristics because the author identified a signature in the participant that drives or that is associated with a better response to the cancer immunotherapy using anti-PD-1. We can also observe that gut microbiome can affect the response variation. In this case, all individuals respond to the drug, but with a lower or a greater extent. And this is also partly due to a variation in their microbiome characteristics. Finally, we have observed that microbiome can really affect the safety of certain drugs. This is the case for the chemotherapy with irinotecan, in which we observe that some gut microbes can reactivate the drugs into the gut, and this leads to severe diarrhea and complications for the participants. Then gut microbes can also change the pharmacokinetic and pharmacodynamic of some drugs and affect the efficacy by inhibiting or activating these drugs. So microbiome appear as a new component to take into consideration in the risk analysis during the drug development process, and this This result is due to the emergence of omics technologies. The third application concerns the evaluation of what drug candidates impact on the gut microbiome composition and function, because here again, several studies have demonstrated that some drugs can really alter and change the composition and function of the gut microbiome.
Yeah.
And this can have consequences for the human health, because I told during the introduction that some microbial metabolites produced during this acceleration can regulate some key physiological function for the host. So one example is the metformin. So in the case of the metformin leads to an increase of two main bacteria that are Akkermansia muscipula and Escherichia coli. In the case of Akkermansia muscipula, the increase is associated with beneficial effects of metformin for the host. But in the case of the increase of Escherichia coli, we observe some gastrointestinal discomfort and side effects associated with the use of metformin, and that is partly mediated by the gut microbes. So here again, microbiome is a new component to consider in the risk of analysis of the drug developments. So finally, I think you have all understood that omics technologies is an association of very difficult step of analysis, and the workflow of analysis is really complicated to interpret because it requires a multitude of several different skills, such as skill in molecular biology, to be able to isolate DNA, for instance, from the samples. But we also need several skills in bioinformatics or in biostatistics. And it's very difficult to find people able to analyze the entire workflow of analysis for omics from the beginning to the end. Another limitation is that each stage of this workflow of analysis can be gauged, because for each step we have several choices. For example, for the DNA extraction, we have on the market different kits for isolating the DNA from different samples, and this can change the efficiency in the DNA isolation, and for consequence, this can change the results of the analysis. We can also have some gains in bioinformatics, because we have different tools and software for pre-processing the sequence read, and the same limitation is present for biostatistics parts. So we really need standardization, we really need more standard or reference material to implement in each state in order to control the reliability and the reproducibility of the data. Finally, you need to remember one slide presented during the introduction, that microbiome is an organ under a constant remodeling in response to several factors. And this is problematic because we really need to control and to know every confronting factors that can appear in our studies and that can change and alter the microbiome composition and function to perform the analysis. So in conclusion, omics opened the door to the big data in microbiome field and gives the access to a multitude of applications. The first one is the identification of new drugs and candidates linked with microbiome. The second one is the integration of microbiome as a new component in the benefit-risk ratio during a drug development. And the third one is that omics allow to move towards a personalized medicine if we are able to standardize all the different steps in the workflow of analysis. So I would like to thank for your attention. Please feel free to contact us if you have any question, and there will be a workshop discussing the different applications this afternoon. Thank you all.
offered. Now I would like to invite Professor Kostas Demesos, who will present the topic of new health technologies.
So good morning. Good morning, everyone. I'm Professor Kostas Demesos from the University of Athens, Department of Pharmacy. And I belong to another scientific field, and I will focus on complexity and nanotechnology. First, I would like to thank the organizers for inviting me to deliver a lecture on this topic. And I think that nanotechnology is a challenging field and that provide us tools to produce new medicines, encapsulating bioactive molecules to nanoparticulate systems. We live in complex times, and Stephen Hawking has called this century the century of complexity. In 2021, three scientists received the Nobel Prize in physics for their work on complex systems. The questions that have been raised are how shall we define a complex system? And what is the relationship between emergence and complexity? How shall we identify causal relationship in complex systems? All these questions have received a great deal of attention in science philosophy, and the relationship between causation and complexity remains to be understood. A complex system is a system which contains many independent constituents which interact non-linearly and has numerous interconnections and interactions which are difficult to describe, understand, predict, manage, design, or change. To study complex systems is a challenging field, and we have to keep in mind that we have to identify the small system. Which is a small system? There is a huge area between bulk material and the molecules from which it is consisted. So this area is named as a mesoscopic area, and this mesoscopic area includes small complex systems. I have to point out that small systems differ from those in macrocosmos, meaning that the cosmos that we are living, and deviate from the classical thermodynamics and biophysics. It is obvious that nature is complex and is related to the morphology of the biological entities, and the role of morphology plays a crucial role for producing innovative medicines, and we have to keep in mind that by producing nanotechnological entities, we have to take care about the morphology and the complexity. It is well known that proteins are complex systems. They are composed of amino acids. The sequence and the conformation properties affect the functionality of proteins. You have in mind that spike protein regarding virus SARS-CoV-2 is a complex protein, and the sequence of amino acids affect its functionality. This slide nicely presents a complex artificial nanoscale drug system, and that are composed of tetramers or structures that are defined as nanoforms. We can see the cavities and the pores, the complexity of the internal morphology. These cavities and pores can accommodate bioactive molecules. So I would like now to share with you the main difference between a complicated system and a complex system. A complicated system, like a car engine, is composed of materials.
the materials can self-assemble in order to produce a gas engine. On the other hand, if we have ant colony, we have ants that by using a communication network can self-assemble and produce complex and functional structure, providing new morphologies depending on the environmental conditions. So this is the main difference between complicated and the complex system, the self-assembly process. Can you see the video? So, okay, here is phospholipids. You can see phospholipids that are the main constituents of our cell membranes. They are composed of a polar chemical group and they are composed of a polar group and hydrophobic and the hydrophobic tail. They can come close to each other and produce microdomains that can produce huge domains that correspond to cell membranes. So this is a self-assembly process that facilitates the production of artificial cell membranes that can accommodate bioactive molecules. Okay, the most important thing in such self-assembly nanostructures and complex nanostructures is the internal morphology in order to be promoted as new areas. For example, here we have 3 cubes. The long range order is the same, is cubic, but the internal morphology, meaning the short range order, is different. So this short range order can affect the stability of the system, the encapsulation efficiency, or the release profile of the encapsulated drug. So the internal morphology plays a crucial role. And we have 3 layers of complexity. The first one is the internal complexity of a system, of a complex system. The second one is the external complexity, and more importantly, the interface complexity between internal complexity and external complexity of the system. And there are several metrics that we can use in order to identify the level of complexity of the system. For example, metrics should be done on functions and on the variables of the system. We have to keep in mind that the thermodynamics of the systems play a crucial role in its survival. For example, thermodynamic entropy corresponds to Boltzmann-Gibbs entropy, entropy von Neumann corresponds to quantum entropy, and entropy Shannon corresponds to information entropy. So all these entropies and more other should be taken into consideration during the evaluation process of a complex system. And the question that has been raised is the complexity of a nanoparticulate system is the barrier for their or for its reproducibility? For example, nanosimilars. Nanosimilars are products like generic products that are copies of the prototype. Up now, there are not such nanosimilars in the market due to the complexity of such structure. So, do the non-equilibrium and quantum effects play an important role in the production identical or statistically similar copies of the prototypes? So this question received a great deal of attention in the community, and the relationship between complexity and reproducibility regarding copies of complex medicines remains to be understood. There are several publications in the literature combining complexity and nanotechnology, and I suggest to find some of these reference for study. And another question is there any relationship between nanodimension and complexity? A short introduction concerns nanotechnology, which is the engineering and manufacture of materials at the atomic and molecular scale and refers to structure, probably in the 1 to 100 nanometer size. And here is the scale of nanoparticles. Some of the main types of nanosystems, like liposomes, polymeric micelles, dendrimer, and some other important structures that can accommodate into the interior part bioactive molecules and transport them to the target tissues, increasing their solubility, increasing the targeting process, reducing the toxicity, increasing the physicochemical stability of the encapsulated molecule, control the drug release, and all these are biocompatible and biodegradable. I have to share with you these nice slides which presents three types of medicines. The first one is the small molecule drugs. These small molecule drugs have low molecular weight, their structure is simple and well defined, they are more or less stable, and more interesting, they can produce identical copies. On the other hand, we have compounds. Okay. Biocompounds and non-biological complex medicines. The most important thing here is that these two types of medicines are very complex systems, and it's difficult to reproduce identical copies of these compounds. Examples of complex nanosystems are there are already in market compounds, medicines like anti-cancer drugs or additional whatever. We have to keep in mind complexity, morphology, and functionality during the preparation process. Liposomes are important and well-known structures that can incorporate bioactive molecules, and cholesterol is one of the compounds from which it consists. And a liposome, they are considered as pseudospherical vesicles, and stealth liposomal technology is one of the most often used in liposomes. I remind you that Pfizer vaccine has stealth properties because the vaccine decorated with polyethylene glycol on the surface in order to avoid the response of the immune system. So stealth properties are very important. Liquid crystal is another class of nanotechnological entities that have high complexity, and the morphology is related to the molecular order and affects their functionality. Some photos, some pictures from my lab, we can see lipid nanoparticles or liposome and liquid crystals that are used in order to incorporate bioactive molecules. And here you can see the complexity, the internal complexity. There are many channels that can accommodate proteins, so genetic material, or small drugs. And you can see that the surface characteristics are different. So it is difficult to reproduce identical products, and this is a barrier in order to produce, for example, copies of the vaccines, especially those against SARS-CoV-2. So some other figures from my lab received by using cryo transmission electron microscopy. These are cubic liquid crystals, and you can see also the internal morphology and the complexity. These cryo-10 figures correspond to lipid nanoparticles that can deliver mRNA, and the morphologies depend on the nature of biomaterials that were used. So we can see that the morphological characteristics are not identical. And this is due to the complexity of such systems. Some other photos presenting the complexity and the internal complexity, and moving on to the SARS-CoV-2. SARS-CoV-2 is composed of the genetic material, which is in the interior part, and the proteins, the most important protein is the spike protein, which can interact with receptors on the healthy cells and promote the infection. sorry. SARS-CoV-2, the virus, belongs to complex systems, and its evolution and mutation must be observed under the lens of the nonlinearity as they are far from the equilibrium condition. Recently, we published a paper in the Proceedings of European Academy of Science and Arts, in which we argue that SARS-CoV-2 is a complex system, and the vaccines that currently are in market are also complex systems. Okay, here is a slide which presents the difference between a liposome, which is well-known nanostructure, and lipid nanoparticles, which are used for producing carriers. 5 minutes, okay, produce nanoparticles that can incorporate genetic material. The difference between liposomes and lipid nanoparticles that were used for producing vaccines against SARS-CoV-2 is the internal morphology and the internal complexity. We can see in lipid nanoparticles, there are different domains that can interact with the genetic material, transport this genetic material to the cells. Okay, Novavax produces a vaccine based on the new technology which is named recombinant nanotechnology. This technology based on the concept of pleiotropism. And recently I published my thoughts regarding pleiotropism in LinkedIn, but I would like to go into details because I have no time. All these lipid cells and the concept of pleiotropism, of self-assembly, and nanotechnology are the main issues that we use in order to produce vaccines against COVID-19. And in my view, there are two main directions. The first one is genetic approach, and the other one is nanotechnology. Genetic approach, due to the pandemic, emerged research directions in more genetic approach, but we have to keep in mind that complexity, morphology, and functionality should be taken into consideration during the production process of such complex systems. And in my view, I think that artificial intelligence in the pharmaceutical industry should be taken into consideration in order to facilitate the identification of the internal morphology of such complicated systems. Artificial intelligence with nanotechnology will assist in the facilitation, optimization of the development of nanomaterials and nanostructures. And it is important that we have to educate not only scientists, the students, but also the society, and social engagement in research and innovation is a challenge. So I propose two main tools, responsive research, innovation, and STEM, among others, that are very promising in order to educate the students, scientists, and of course society. And now, coming to the end, I would like to show you two slides in order to combine nanotechnology, nanoscale structures without the beauty. Gustav Menzer, who was a German artist and a political activist, who developed the concept of auto-destructive art and the art strike. He initiated destruction art symposium in 1966 and used the concept of pleiotropism and liquid crystalline state of matter. You can see here in the slide paintings that contain nanostructure and scatter light in different ways, produce different colors, and correspond to art. So the art and beauty at nanoscale is an amazing proposal for the scientists, and here is another imaginary large scale of beauty and complexity. complexity, and you can see dendritic nanostructures by the artist Angela Pratt, who admires the art and beauty at nanoscale. You can see dendrimers. Dendrimers are nanoparticulate systems that are used in order to encapsulate bioactive compounds. I student, okay, thank you very much. And here is a recently published book which presents the conversation between the invisible world, the microcosmos, the nanocosmos, and the macrocosmos, the cosmos that we are living. And I think it's an interesting book. So thank you very much. I'm on time. Thank you very much for coming.
Thank you, Professor, for your presentation. Now I would like to give the floor to Dr. Florence Gender Blackman from Dakota.
Okay, thank you so much. I'm going to try a new area of the room, but thank you all for having me. I'm very excited to be here today. So I'm going to talk about personalized medicine and its value and implementation in cancer care. So today we'll go over an NCODA introduction, a personalized medicine overview, genomics and cancer, and then some ways that we can implement. So as a brief introduction of NCODA, we're a grassroots, not-for-profit international organization, and we're committed to developing unique resources to help medically integrated oncology teams deliver top-tier care. So we have a little over 6,000 members internationally. Many are based in the US. We have a lot of pharmacists, pharmacy technicians, nurses, physicians, and we actually have over 50 student chapters now. And I came to our team, I've been with us for a couple of years, and I was previously a pharmacist in an oncology practice in Florida. So personalized medicine has become so important in cancer care. So to start, I would like to go over a few of the hallmarks of cancer. So just as a reminder, there are several processes that can lead to cancer. Normal cells evolve into a neoplastic state. There is successive acquisition of hallmark capabilities, and it's a multi-step process of tumor pathogenesis. So not just one thing that leads to cancer, there are many, many steps that arise along the way. So there are various treatment options for cancer. There are lots of, we have kind of our basic methods of chemotherapy. So if you see on the screen from the left to the right, as you get more to the right, targeted therapies are becoming more common than just our standard chemotherapy, one-size-fits-all approach. So the definition of personalized medicine, you can see two different definitions on your screen. But basically, it's taking an individual's genetic information and it's using that to base treatment and to hopefully improve outcomes for patients. So why personalized medicine? So why is it better than our standard traditional medicine? So as a reminder, traditional medicine really takes a one-size-fits-all approach. It can lead to variable effects from patient to patient, and it has potential for less efficacy and or more toxicity. So we're giving patients the same treatment no matter what their genetic makeup is. And it really, you may have some patients that chemotherapy works wonderfully and some patients that it may not respond at all. And then personalized medicine, so why is this better? It is going to take into each individual into account. It really tailors the treatment towards the patient. We can avoid using unnecessary therapies. We can adjust the dosing, and it optimizes optimal outcomes. So genomics in cancer. Just as a reminder, the definition of pharmacogenomics in cancer is a component of genomic medicine that involves using a patient's genomic information to tailor the selection of drugs used in their medical management. So again, we're using their personalized information to really tailor their treatment. So as a reminder, there are somatic and germline variants. Somatic variants are tumor DNA. They're not inherited or transmitted to offspring. And then germline are in the host DNA, and they are often inherited and transmitted to offspring. So really, in most of cancer treatment, we're going to be looking at the somatic mutations. And in that, we also have passenger and driver mutations. So there are two types of these mutations. The passenger is a variant that doesn't influence a cell's growth, so it generally does not determine prognostic or predictive values, and we'll talk about those in a couple of slides. And a driver, those are the ones that we're really going to be concerned with in cancer treatment. The driver is a barrier that confers a selective growth advantage, and it's the most clinically relevant in determining prognostic or predictive value. So again, when we're treating patients, we're really going to be concerned with these driver mutations. So biomarkers, what is a biomarker? According to the European Federation of Pharmaceutical Industries, it's a biological characteristic of the body that can be objectively measured and quantified. So essentially, it's any gene, molecule, or characteristic derived from tissues or bodily fluids. In oncology, the biomarkers are abnormalities or mutations that are found in cancer cells. And then we have, as I mentioned a couple of slides ago, we have the predictive or prognostic biomarkers. Predictive provides information regarding response to a specific treatment. So for example, EGFR alterations are assessed at diagnosis of metastatic lung cancer. And in the workshop later today, we will discuss one of the therapies that you can use for patients that's based on this EGFR mutation. And then we have prognostic, and those provide information about cancer outcomes, such as progression-free survival or overall survival. So that's when a patient may have a specific mutation, and that's really going to determine, how well their cancer is going to progress. So are they going to respond to treatment? Is it a worse outcome in the end? There are different types of sequencing, which you can see on the screen. The next generation sequencing, it is the gold standard. It's preferred because it can assess multiple gene alterations, and you can see the process again on the screen. So implementation, what does all of this mean, and how are we using it in practice? There are many guidelines. So after a patient receives sequencing, where do we go? We can use these guidelines. The Clinical Pharmacogenetics Implementation Consortium is an international consortium of individual volunteers and a small dedicated staff, and they want to facilitate the use of pharmacogenomic tests for patient care. So their goal is really to address barriers to clinical implementation and pharmacogenetic testing. They create freely available, peer-reviewed, evidence-based, updatable, and detailed gene and drug clinical practice guidelines. And the website is on the screen, so I would encourage you to check out their website as well. And then INCODA, so our organization also develops many guidelines. We call them positive quality interventions, or PQIs. They are peer-reviewed clinical guidance documents for healthcare professionals. They foster better care for patients through appropriate patient identification, treatment selection, increased speed to therapy, reduced costs and hospitalization, and improved adherence techniques. And you can find those on our website. We have them for a lot of drugs, a lot of disease states, and when I was in clinical practice, we found these very valuable for training other employees, just a quick reference guide. So instead of a 30-page PI that you have to read that's in tiny print, it's a 2 or 3-page reference. And the workshop later today, we will go through a little more details of personalized medicine, and we'll take a few of those PQIs, walk through them, talk about how you as a pharmacist can use them in clinical practice. And INCODA membership is complimentary for all healthcare providers that are in oncology or students. So if you do have an interest in oncology, I would encourage you to join. And also, I'm happy to chat with anyone. So thank you so much, and I will turn it back over to you.
Thank you, Lois. like to give the floor to our next speaker, Doctor of Pharmacy Perspective Studies, and also the R&D Director from Apple.
So, good morning from me. Thank you very, very much for the invitation. It's always an honor and a pleasure for me to speak in front of pharmacists. I'm a pharmacist myself, having spent lots of years of my life at the pharmacy school, mainly of Athens, under the tutoring and the supervision of my mentor, Professor Konstantinos Demedjos. And it's very nice, very convenient for me actually to be speaking almost right after the talk of Professor Demedjos, as I can skip a little bit of the introduction. So I've done a master's degree and a PhD focusing on pharmaceutical nanotechnology. And I'm going to present you the more, let's say, industrial and kind of fun part of this process, always connected with omic technologies. That is the topic of this current conference. So I've been working in this company called Pivita. It's a natural products company. Oh, sorry. For Greek students should be aware of the company. For the ones that don't know it, it's a natural products company, mainly natural cosmetics, also food supplements and foods as a lower part of the business in terms of development and turnover, mainly relying on Greek nature, on the bee society, and the derivatives of bee society in pharmaceutical and aromatic plants. And this is a keyword in the initial slide of the company, raised by science. We use a lot of contemporary technologies in order to design, research, a little bit at the fundamental stage, and of course a lot more at an applied research level, and then develop final products, but also the raw materials, the bioactive raw materials that are incorporated in final products, which is actually the innovative and the very interesting part, at least for the pharmacist that has dealt with pharmacology and pharmaceutical technology thoroughly. So the brand was created in 1972 by a couple of pharmacists who, in their pharmacy laboratories, used to do, let's say, pretty novel mixtures for that time, mixing essential oils, big products into producing their final cosmetic products that were going actually pretty well. So they started the company in 1979. It's one of the pioneers globally, actually, in the field of natural cosmetics. And now it's a very big corporation. I will show you some slides afterwards showing the international activity of the brand, which is called Vita, as I told you, which means the word of the bee, Avis, bee in Latin, and Vita. So the raw materials of interest are the bee products: honey, broccoli, beeswax, royal jelly, mainly, a bit pollen also. And the other part is organic Greek, okay, more big broader term, organic Mediterranean nature. So let's get to the point of the talk. I won't focus on of course, the various families of bioactive compounds in both bee products and pharmaceutical plants. I will just focus on one category, which is the polyphenols. So you should be aware of them. They're very interesting for cosmetology because of various mechanisms of bioactivity and interaction with skin cells. And of course, as most of natural products, they come with a bunch of, let's say, not difficulties, but actually challenges for the formulator, for the pharmacist working in research and development laboratory, which are always solubility, stability, which is a very, very main issue, very, very easily biodegradable, bioavailability, and release rate. When we're talking about the skin, the term is not exactly bioavailability, though it is described like that. Also, the bioavailability refers to plasma concentrations of a certain drug. Here we mean that we need these molecules to be in contact at the right place to reach their target for long enough time in order to exceed their bioactivity outcome. So there are many strategies in order to tackle around these challenges, which may not be so severe, especially the stability issues or the solubility, for example, working in an academic environment, but working for the industry, this is absolutely crucial because imagine you produce something that contains polyphenols that will be the main bioactive ingredient that get oxidized when being inside an emulsion after a couple of months. So there are many strategies in order to overcome those. What is very interesting to me and what I have inherited from the Nano Lab of the Pharmacy School of Athens is delivery systems that we have developed and customized for the skin tissue, and from the various categories. We are focusing on lipid systems and carbohydrate-based systems that, of course, have many different variations depending on the need of the design. From the carbohydrate systems, cyclodextrins are great systems. They're circular molecules derived from starch hydrolysis, very simple molecules actually, but very accurate. That depending on the monomers of dextrin and on the periphery groups, they can have to be categorized in several molecules that have different kinds of solubility and different molecule entrapment capabilities depending on what you want to incorporate those molecules. I will skip, of course, the part of the liposomes because I'm speaking after the master, Professor Revetos, but keeping in mind that the design of liposomal systems came back in the 60s from an idea of biomimicry of the actual skin cell. So taking this awesome membrane mechanism that we have in all our cells and transferring it to those vesicles that can be, of course, a lot custom made in order playing with their physicochemical parameters in order to design systems that can reach specific targets and have specific efficacy interacting with incorporated drug or molecule in general. So, what we combine in the phytochemistry laboratory that deals with these kind of systems that I will show you some case studies later on, with the biochemistry, the phytochemistry lab, with the biochemistry laboratory, it's biochemistry and molecular biology, where we do omics experiments, is actually a combination of technology of new systems incorporating molecules from the Greek nature, mainly, that are assessed in each stage, so from very initial stages of the design until reaching the final product with mainly omic technologies, and among omic technologies, playing a lot with transcriptomics, with epigenomics, and with metagenomics. Of course, we don't have, it's too much for the companies to have sequencing facilities in-house. We outsource this, but these are very, very extremely sensitive and very informative techniques about the future, of course, safety, which is the first of all, and second, efficacy of the product, but not only the efficacy, but the exact mechanism of activity, which is, let's say, a golden information for developers and formulators. So we have built up a gene database working on real-time PCR, so doing transcriptomics, so dividing our human genome, that is of course focusing on the skin, on several mechanisms that can be categorized, let's say, as a translation for marketeers, mainly into those keywords that are very common in cosmetics, so aging, oxidative stress, prevention, inflammation, skin pigmentation, etc., etc. So let's see very briefly some examples of how these things work. So one pattern we have developed kind of recently is a control release system, a double system consisting of interacting liposomes and cyclodextrins with the polyphenols of propolis. So in this process, the polyphenol during extraction, which is very essential because it's a, let's say, one-pot process, which is very crucial for the industry. It is very, very lean, let's say, industrial process. During the extraction, the polyphenols get trapped with the cyclodextrins, and as a sequence, this combination gets trapped and is turned into an impossible system that gives a very, very beautiful release curve for the polyphenols and antioxidant activity. Giving very interesting results in terms of cell metabolism, increasing cell metabolism, or preserving the cell metabolism after UV radiation, both in skin cells and three-dimensional skin tissue experiments, giving exceptional results in maintaining the levels of metalloproteinases, which are enzymes that under stress are overexpressed and degrade mainly collagen and in general the building blocks of the extracellular matrix of the skin. And by transcriptomic experiments, we have seen a great increase in the anti-inflammatory activity compared to control, an extremely significant increase in skin hydration and an increase in the vasodilation of the cells, of the skin cells, and in parallel DNA damage and protein damage significant decrease under UV radiation stress. So all of this you can find both in a couple of publications and a global patent that was awarded to us several years ago. Then we took this process one step further to play with different polyphenols from different materials. So coming up with another patent that combines a burst effect, a burst polyphenol effect from propolis in cyclodextrins, and from Vitis vinifera leaves, some specific varieties of Vitis vinifera in cyclodextrins and liposomes. So as a visual, which is interesting, first of all, the cyclodextrins release propolis in around 30 minutes, and as a second step, the polyphenols from Vitis vinifera get released during 8 hours, giving giving us very interesting release curves and also very interesting release inside human skin cells, as we have seen from the skin penetration studies with tape stripping. So the concept here was the activation of skin defense mechanisms against environmental factors that accelerate aging. We know that UV radiation is the key driver for photoaging. The urban pollution is the driver for inflammation, for skin inflammation when living in an urban environment. So what we did is we took fibroblasts, we bombarded them with particulate matter from urban dust and UVA, and we saw the production of three key biomarkers: collagen 1, collagen 3, collagen, and elastin. And we saw great results in the restoration of collagen 1, collagen 3, and elastin during those studies. So the protective effect of this combinatory system of polyphenols. And also, we did the perspective experiments without stress, so to see what happens under normal conditions in our fibroblasts. And we saw collagen levels decreasing by 70%, and metalprotein levels decreasing, which is also very important, complementing this mechanism of activity. A great decrease in hyaluronase synthase that mediates the production of hyaluronic acid, and it's crucial for hydration. And finally, doing proteomic experiments, we saw elastin levels increasing fourfold, more or less, and collagen levels increasing 129%. So all these results in total reassure us about the effect on the extracellular matrix, on the proteins of extracellular matrix. That is the key target for this development, for this pattern. Also, it's a zero-waste approach, so the leftovers, the extraction of propolyl dextrose becomes this so far. And the vinyl leaves, which is very interesting that they're not cycling ingredient, they are just byproducts of winemaking in Santorini. They are through this process, and the leftovers, the waste, let's say, becomes soil enhancer that gets sent back to Santorini to close the sustainability, let's say, loop. We don't have time for the last invention, so it's a system that looks like the rest of the system, both cyclodextrins and and liposomes incorporating royal jelly. So what's the issue with royal jelly? We need to keep it in the fridge. Once we open it, we consume in very few days because it's very, very easily biodegradable, especially the very beneficial lipids and the protein molecules. So what we did is we actually did a system in order to preserve the freshness of Greek fresh royal jelly throughout the lifespan of the final product where this bioactive material gets incorporated. So we have this very nice release here for as a biomarker. We use the main bioactive ingredient, then HDA. This main of the royal jelly, with the respective transcriptomic experiments going very well, and especially extremely good results in indofuscin 2, which is an indicator of mitochondrial fusion that promotes to a very large extent to the increase of cell energy metabolism. So you can also find the publications and the patents online if you want to dig more into the mechanism and how all these systems work. And as an overview, I would like to state that you are very, very lucky because now you're initiating your career in front of the dramatic breakthrough in, of course, atomic technologies. That is the topic of this conference, but also surrounded by technology, by personalized medicine, and these are things that are nowadays feasible both at an academic level and in an industrial level to really make an impact on things that have to do with sustainability and healthcare by using very contemporary technologies. So, just to have an overview that these inventions, we are very proud of this because we take those materials from Greece, we give them great value because of operation, just delivery systems and having great efficacy means that you need small quantities of very high added value materials. That is very important for sustainability issues also. And we drive these into 50 active markets around the world, and you can see them in pharmacies around the world or in corners from Europe to Asia. So thank you very much.
Thank you. Thank you to all the speakers for your amazing speeches. And now we'll have a short coffee break, more than 20 minutes. We'll meet here 11:30. So please make sure that you will be on time. And one more thing, please keep in mind that you will need to have your QR code scanned, so prepare it before you will enter. Thank you.